Hello, and welcome to BeOne Biotech, the weekly podcast from Labiotech. I'm Dylan Kassane, and this is episode 172 for the podcast. Cancer is one of the biggest health challenges worldwide. While progress has been made, millions of patients are still in need of new treatment options that better address the complexities of their disease. This week, I'm sitting down with Mark Lanasa, Senior Vice President and Chief Medical Officer, Solid Tumors at BeOne Medicines, a global oncology company focused on developing medicines for some of the most challenging cancers. BeOne's in-house clinical engine, combined with an extensive array of advanced cancer models, is helping to accelerate the discovery and the development of new therapies. We discuss the challenges in cancer treatment and care and why tackling these challenges takes a carefully designed approach. I hope you enjoy my discussion with Mark Lanasa. Mark, welcome to BeOne Biotech.
Thanks very much for having me, Dylan. It's really a pleasure to be here.
Yeah, it's great to have you here. And to kick things off, Mark, why don't you tell me a little bit about yourself and what you do at BeOne?
Sure. Thank you. So I am a physician scientist by training. I have a background in training in medical oncology and hematology. I also have a PhD in genetics and molecular biology. I actually spent about 6.5 years in clinical practice as a medical oncologist, focusing on the care of patients predominantly with leukemia before I made the decision to transition to industry. I have been in industry for a little more than 12 years, working on the sponsor side to try to bring innovative medicines to patients with cancer. This is informed by my experience as a clinician and as a clinical trial investigator. I have been at BeOne Medicines now for a little more than 3.5 years. My role here is I'm the Chief Medical Officer for Solid Tumors. So I lead a global team of physicians and scientists who are designing and delivering global clinical trials for patients with cancer to develop novel therapies for patients with cancer across a spectrum of different solid tumors that we can talk about as we go on. That is the majority of how I spend my time, is working directly with the teams. I also have some other components of my role. It's my pleasure to also be something of a technical ambassador, where I get to travel to medical meetings and meet with clinicians and investigators and visit sites in support of our ongoing development programs.
Now, you said you worked as a clinician. Maybe you can tell me a bit about, the challenges that you saw working when it comes to cancer treatments, and how does this shape what you're doing today?
Yes, so it's becoming further and further in the past than I might like to admit, but I was in clinical practice, say 15 to 20 years ago. And during my time as a clinical trainee in oncology, really the state of the art was largely to give cytotoxic chemotherapy, and sometimes with steroids, but that was really the core of what we did as medical oncologists. As I mentioned, I treated patients with acute and chronic leukemias, and again, largely gave those patients cytotoxic chemotherapies. The good news is that's well justified, because some patients can be cured by chemotherapy. But the really bad news is that it's the minority of patients who are cured with chemotherapy alone. And virtually all patients have chemotherapy-related side effects, some of which can be really difficult. As I briefly mentioned, I also worked as a clinical investigator working on clinical trials, and I had this amazing experience that as I enrolled patients in my clinic on targeted therapies, non-cytotoxic therapies, I saw these amazing responses. Of course, not everyone, but I saw some patients who had dramatic responses to targeted therapies, significant regression of the tumor of whatever cancer type they had, with a much, much better side effect profile than what they were experiencing with the chemotherapy that I was giving as part of routine care. And this was kind of my epiphany moment that I felt, with my scientific background, I thought, this is really what I want to spend my career on. And I made the decision to move from the physician side over to the industry side, again, to design and deliver clinical studies with the intent of developing better and better tolerated treatments for patients with cancer.
I mean, we're talking about cancer, but cancer is not just one thing. There's not just cancer. There's not just even lung cancer or breast cancer. It's pretty highly differentiated.
Absolutely. So this is the challenge and the opportunity that we face in oncology development today. We talk about cancer as a disease, but the biology of cancer is highly diverse across different tumor types. And indeed, even within individual tumor types, there's great biologic diversity. Within the solid tumor organization here at BeOne, we focus on three areas, those being lung cancer, breast and gynecologic cancers, and GI cancers. There's substantial biologic diversity within each of those areas. Uh, Again, since the time when I was a clinician, we have discovered that lung cancer is really an amalgam of different diseases with different drivers. Some patients have mutations in a gene called EGFR or ALK or ROS or others, and those patients are really sensitive to inhibition of that so-called driver mutation, whereas other patients just have an amalgam of different pathways that drive their cancer. We see similar phenomenon across breast cancer where there's different molecular subtypes, and gastrointestinal cancers are really quite different biologically based upon the tumor from which they originate. Liver cancer is entirely different from pancreatic cancer, is entirely different from colorectal cancer.
But it's differentiated, but it's also adaptive. I mean, these cancers, they don't just stay still.
So this is one of the greatest challenges that we face both as clinicians and as researchers, that for the great majority of cancers, the cancer is not driven by a single gene or a single pathway. There are examples of that. For example, chronic myelogenous leukemia responds very well to BCR-ABL inhibitors, but that's really the exception, that the majority of different types of cancers have multiple pathways that are dysregulated, or the cells have acquired multiple different acquired mutations on their pathway to becoming a cancer cell, and that means that they don't follow normal biologic processes in a number of different ways. They grow more than they're supposed to, they don't respect anatomic boundaries, they don't die when they're supposed to. Therefore, it's very difficult to treat cancer with a single modality, and this is why we think combinations are so important, particularly combinations that target different mechanisms related to the cellular biology of cancer.
I guess, like you're saying, with so many different types of cancers, all of these different challenges, the fact that it's adaptive, there's no one size fits all approach. How do you actually approach these diseases and structure your research and development at BeOne?
Thank you. That's a great question. So we do feel that we're taking a slightly different approach from that of other companies. I think what's critically important is, again, having a deep understanding of the biology of these different diseases, that ultimately, as I mentioned, cancer is a disease of acquired abnormalities, some genetic abnormalities that lead to changes in the biology of the cells, and those changes can ultimately be exploited as liabilities of the cancer cell. Again, the cancer cells diversify so that blocking any one pathway may not be enough. But through really structured and thoughtful research, we can understand what combination of pathway inhibition might lead to a best approach for an individual patient or a certain type of cancer. So we're very focused on a couple of different things related to that overview of cancer biology. So one is that we think that it's important to have a relatively large portfolio of investigational molecules. This enables us to cover lots of different pathways, enables us to develop across these biologically distinct types of cancer. And again, it enables us to have combinations. And therefore, we need to bring a fair number of new molecules into the clinic each year to continue to diversify and grow our opportunity space. So again, this is all based upon having a research organization with a deep understanding of biology, the capabilities of developing new therapies that can modify the biology in a way that we think is likely to bring benefit to patients.
You say you're working on multiple fronts there. How many assets are we really talking about?
So it has been a really exciting time during the 3.5 years that I've been at BeOne Medicines. Today, we have approximately 40 different molecules that are either investigational or approved. We have gone from 6 to 8 molecules in solid tumors when I joined to now approaching 30. So there's been a tremendous expansion of the pipeline. Now, we brought 13 investigational medicines into the clinic in 2024. We believe that this is the most of any company in any therapeutic area. But it's also important to understand that that's 13 out of a denominator of up to 80 molecules. So we have up to 80 preclinical programs at any time. So we actually do have a very high level of stringency for what we actually advance into clinical investigation.
And you're talking about bringing all of these assets up and so many at the same time. How fast are you moving? How are you moving so quickly?
So this is where our unique research and discovery organization interacts with our unique development organization that I'm a part of. So a philosophy is that to work with urgency to address the needs that patients with cancer have, that we've made a decision to really have the entire drug development ecosystem to be in-house. So I've referred to our in-house research and discovery organization, which includes over 1,100 PhD scientists. We have in-house manufacturing, so we largely manufacture our own investigational as well as approved medicines. And then we have in-house development, so myself in clinical development, but also regulatory affairs and clinical operations and patient safety. And this enables a couple of things. By having that in-house ecosystem, that enables us to make decisions quickly, to move faster, by not having an intermediary, a contract research organization working with sites. It also enables us to control costs, which we then intend to pass on to patients and enable global patient access. So we think that having this wholly in-house model has advantages both in terms of speed and speed and cost. And then importantly, as I alluded to briefly before, we think that investigators also like it because they're working directly with us. So again, I go and visit sites, I talk to the investigators. If they have questions, if they have concerns, they know who to reach out to, and we aspire to be very responsive.
So you're doing everything in-house then, the different cancer models, in vitro, ex vivo, that's all in-house?
It is. So again, our large research organization is working from the very earliest parts of understanding cancer biology. They have in vitro models where they'll test an investigational compound that may not even be in the clinic yet, and so-called biochemical assays where they'll evaluate whether it can inhibit an enzyme. Then that might graduate to what's called an ex vivo model, such as a patient-derived tumor specimen that's being grown in a cell culture facility to see if that molecule can then slow the growth of a tumor. It can also look at so-called in vivo models, where we bring those models out of the petri dish and into an animal like a mouse, again, to show if we can block regression of, if we can block growth of the tumor, indeed lead to tumor regression or elimination.
You know, this is really interesting to me because I've talked to other top pharma companies and they don't all work like this. Your strategy seems a little bit different to the way that R&D is normally done.
We believe that it is. One of the things I've heard from our research organization is this relentless commitment to make it better. So there are, again, our 3 disease areas. Within those disease areas, we try to identify targets and pathways that we believe are very important to cancer biology, and then we just relentlessly work on those targets. So one thing that we do that's a little bit unusual is we might bring multiple drugs forward going after the same target because we think, again, that can maximize patient benefit. A good example of that on the hematology side might be that we have an approved BTK inhibitor called zanubrutinib, but because we're trying to make it better, we now also have an investigational BTK degrader, which has recently entered phase 3 studies. And we think those molecules can be quite complementary for a patient with B-cell leukemia or lymphoma as part of their overall journey.
So it's more than just trying to be efficient. You're actually testing things in parallel and finding complementary treatments there.
Exactly. So we also recognize that cancer development is hard insofar as you can have a really good idea that ultimately doesn't work out for reasons related to the complexity of the biology, incomplete understanding of certain models. And therefore, we think this model of advancing multiple candidates But then when we get to clinical investigation, having very clear objectives in terms of what is the PK of the molecule, what is the safety of the molecule, what is the efficacy ultimately of the molecule, having very clear success criteria enables us to quickly evaluate a relatively large number of investigational molecules.
Any sort of clinical development is just really complex, but maybe especially so when it comes to cancer. What sort of progress do you feel like you're making? Do you feel like you're actually moving forward with that clinical development?
Yes, we are proud of what we've accomplished to date. As I've briefly alluded to, we do have two globally approved medicines, zanubrutinib, which is a BTK inhibitor, and tislelizumab, which is a PD-1 inhibitor. We have a third investigational medicine called sonrotoclax. It's a BCL2 inhibitor where we've announced positive top-line data that we believe will enable global regulatory filings. But importantly, as we scaled out our research organization, we've now scaled out the development organization. As I mentioned, we brought 13 molecules into the clinic last year. It's our ambition to bring 8 to 10 new molecules per year into the clinic across hematology, solid tumors, and immunology or inflammation. Another way to think about that is 2 to 3 new molecules per year in lung, breast, and GI cancers. So we do think that we are making an impact, but we really think that this is just the beginning of the story as the full capabilities of the research and development organization come to bear.
And BeOne is still relatively young as well for a research company.
Yes, so we have been around now for 15 years. We were founded in 2010. I think that to the credit of our founders, we have had the same senior leadership team and founders since the time that the organization was created. That has given us, shall we say, a North Star in terms of our objectives, in terms of scientific innovation, and bettering outcomes for patients with cancer around the world. But a lot of the build out that I've been talking about really has happened since, say, 2019 and 2020. We now think that we're at a steady state of we can bring 10 investigational medicines into the clinic each year. So we believe that there's so much more that we'll be able to accomplish over the next 5 to 10 years.
So, Mark, you've got so much stuff that you're looking into at BeOne, and I know you've got these 3 focus areas. Maybe you could tell me a little bit about some of the stuff that's still at the earlier stage, the investigational stage, in these 3 main areas that BeOne looks at.
Absolutely. So we're very excited about how the internal early portfolio is advancing, and I want to be careful that I'm not making claims that something is definitely working, but rather we're seeing evidence that we're meeting some of those specific hypotheses in terms of PK and safety and efficacy that we think would be required to advance an investigational molecule into a registrational intent study. And maybe I can speak to one from each of our 3 focused disease areas. In our breast cancer franchise, we have been very focused on molecules that inhibit cell growth, so-called cyclin-dependent kinase molecules. We have a CDK4 selective inhibitor. This has been, shall we say, something that we view as potential cornerstone molecule in our breast development area because CDK4/6 inhibitors are a foundational molecule for the treatment of patients, of women with hormone receptor positive breast cancer. By having a more selective CDK4 inhibitor, we believe that that might enable a better safety profile while preserving or potentially even improving efficacy over current available therapies. This molecule is a great example of our development capabilities that while we have only been in the clinic for approximately 20 months with this molecule, we have already enrolled over 400 patients around the world, literally on 5 different continents to date. So we have made tremendous progress in describing the early PK safety and efficacy of the molecule, and we think that that's shaping up favorably to move into later phase investigation. Within our lung disease area, we're very excited about what's called an MTA cooperative PRMT5 inhibitor. PRMT5 is a specific enzyme that targets patients who have an acquired genetic abnormality or a deletion of a gene called MTAP. Patients who have a deletion of MTAP, if you then block PRMT5, that blocks certain pathways related to protein synthesis that are necessary for cellular survival, and the cells can then die. So this is present actually in somewhere approximately 15% of all patients with cancer. It's present in a similar proportion of patients with lung cancer. We are excited about the preclinical characteristics of our molecule in terms of potency, selectivity, but it's also designed to penetrate the brain, which is very important in lung cancer, where unfortunately brain metastases are common. And we're very happy with what we're seeing in terms of early safety and efficacy data. And we're looking to accelerate that program, which has only been in the clinic for 8 months. And maybe the last molecule that I'll mention is an ADC, an antibody drug conjugate. This molecule targets a gene called FGFR2b. FGFR2b is an important target in gastric cancer, but it's a tricky target because the natural biology is involved in multiple signaling processes in different cell types, including the cornea, the surface of the eye. And if it's overly inhibited, that can actually lead to visual side effects, which are very, very difficult for patients to tolerate. So by understanding that biology, we have designed a molecule that doesn't inhibit that biology, but can still target that molecule to deliver a chemotherapy-like molecule. So we're very happy that to date, we are starting to see some evidence of efficacy, but we're not seeing side effects that are impacting the eye. So this molecule, again, while it's early days, is working as designed, and that's always very gratifying thing to see when the understanding of the preclinical biology is beginning to emerge as being consistent with what we're observing in the patients in the clinic.
So you've got a couple of medicines already commercialized. You've got these other ones lining up and deep in the clinic as well. What's key to actually continuing to push that forward then?
So in addition to the understanding of the biology, we think that using the latest targeting techniques or modalities is really important to continuing to advance the field. So of the new modalities, the targeted therapies, the protein biologics, we think that a lot of what can be accomplished with those modalities has been accomplished. And therefore, there's a transition that's ongoing to newer targeting modalities. So there's been a lot said and a lot of impact related to ADCs, antibody drug conjugates. These are protein biologics that have a chemotherapy-like molecule attached to them. So it essentially gives much greater targeting specificity to a chemotherapy molecule. This has had substantial impact in a number of different tumor types. But we think that there are other new modalities that we're looking to leverage that can bring more benefit to patients. One that I'm excited about is our small molecule degrader platform. So we call those CDACs. Those are small molecules that are orally bioavailable, or a pill that a patient would take, and they work differently from other small molecules in that they actually break down a target protein rather than inhibiting the target protein. And this has certain advantages, and we think we're just scratching the surface of what can be accomplished with degraders as a new therapeutic modality.
So when you're doing everything in-house, then I guess that means you don't have to focus just on one modality, and you can leave a little bit of room for flexibility. You've got everybody working in the same direction.
Yes. So again, it comes down to having this, shall we say, hierarchical thinking in the research organization, which is to say, first we set the priority in terms of tumor types, lung, breast, GI. Then we have the scientists go to work on identifying targets and pathways that are relevant in those individual tumor types. And then once we've prioritized the target, we then think about, well, of these new modalities, how can we leverage new technologies to maximize the inhibition of the pathway or hitting the target, and how can that translate into increasing patient benefit? So again, I'm very excited about the opportunity that degraders present because their mechanism of action is catalytic, which is to say that one molecule can lead to the breakdown of potentially thousands of copies of protein, whereas an inhibitor one molecule blocks one copy of protein. We also think that the ADC field can continue to evolve. So you could use the same conceptual approach where you have a targeted biologic, but rather than attaching a chemotherapy molecule to it, you could attach something else. You could attach a degrader molecule. You could attach a small molecule that might have systemic toxicities that would make it not a great molecule to take by mouth and have it go everywhere. But if you could give it in a very targeted fashion, might allow us to drug some of the remaining what are considered undruggable targets.
Looking ahead, how is this model going to keep evolving, and what's the potential impact, do you think?
So we're very excited by what the future holds for novel therapies in oncology. It has been one of the great joys of my personal career to see how oncology has transformed from, again, when I was training, chemotherapy and steroids to all of the biologics, targeted therapies, immunotherapies that clinicians now can bring to bear. That said, our work is not complete. The reason why we have prioritized these three tumor types is that they're still high unmet need. We think that there's a lot more that can be done. So we think that our model of having this in-house deep understanding of biology and, shall we say, flexibility in terms of leveraging different technologies will allow us to address lots of different potential new treatment modalities. By having a focus within disease areas and complementary pathways, this allows us to combine investigational therapeutics in a really rational way that's likely to maximize benefit for patients. And again, we think it's just the beginning of what we can accomplish now that we have gotten to the scale that we aspire. We currently have over 100 active clinical trials being delivered in approaching 50 countries globally. So we think that this in-house global model can bring a lot of potential benefit to patients.
I guess cancer R&D, it's always going to be challenging, but it seems like you're at least making some progress. And I mean, working in so many countries and running so many trials, you really have to feel like you're moving forward.
It is. It's gratifying and it's exciting. The scope of the problem is quite large, but again, with the long view, you can see how we're chipping away at it. We would love to have the next major advance come soon. We are all working with urgency. It would be hard to find someone whose life has not been touched in some way by a friend, a relative who has had cancer. So, we're committed to innovation. We're committed to helping patients around the world. And as our motto goes, cancer has no borders, and neither do we.
Perfect. Mark, it's been great to have you here sharing your insights and also learning a little bit more about what BeOne is doing and how you're challenging the status quo really to bring this cancer treatment forward. If people want to learn a little bit more about BeOne's efforts and the pipeline that you've got moving, where's the best place for them to go?
Thank you again, Dylan. The best place to learn more about BeOne is at our website. That's beonemedicines.com. That's BeOne medicines.com, all one word. There you can find detailed information about our pipeline, our approach, and the teams who are driving our innovation. You can also find us on LinkedIn, on X, Instagram, and Facebook, where we'll share relevant updates and news and also insights into overall cancer biology and progress in the treatment of patients with cancer.
Mark, it's been a pleasure having you here. Thanks for being on BeOne Biotech today.
It has absolutely been my pleasure. Thanks for inviting me.
That's it for this episode. Don't forget to check out all the latest news and analysis at Labiotech.eu. I hope wherever you are in the world, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another BeOne Biotech.